Welcome back to this course on organic chemistry in biology and drug development. Last time we have discussed the chemistry of hypertension and what is one of the cause of humoral cause for hypertension, and that is the production of angiotensin II, and then we have discussed how the drugs like captopril, enalapril and these type of drugs were discovered based on the drug design principle, based on the mapping of the active site, and ultimately we got to know that how this the drug can be designed rationally. In fact, captopril is known as the first rationally designed drug. So, today we will change the topic and go to a very important one, which is affecting us all over the world. Hypertension is also like that, but hypertension is usually affecting the people with more aged people than it is not prevalent in younger ones. However, these antimicrobial drugs are very important because throughout our life, some point or other, starting from the childhood, that we suffer from infections that are caused by small microorganisms. The microorganisms are small because the name suggests that, and these microorganisms include bacteria, it includes virus, it includes fungus, all these. Okay. So, we will discuss the antimicrobial drugs and their chemistry and how actually they evolved to save the human lives. Okay. So, antimicrobial drugs are basically chemicals which work against microorganism infections by a microorganism. Okay. Now, the story started like this. Every topic of research has a history behind it. So, drug discovery process, especially the antimicrobial agents, that research started, and it has a checkered history behind it. Okay. It was first Louis Pasteur. He believed one idea that one microorganism survives against the attack of other microorganisms by producing some chemicals. It is like the survival strategy of our country, that we produce different weapons or procure different weapons, and that is to protect our own country against the invaders. Similarly, in the bacterial world, one bacteria wants to prevail on the other. Okay. So, if to prevent that, the bacteria have evolved a mechanism by which they produce molecules surrounding them, and these molecules basically can kill other microbes surrounding it. Okay. So, that is the defense mechanism of a microorganism like bacteria. So, that was Pasteur's theory. However, when Pasteur attempted this because to isolate some of these chemicals, he found that these are also toxic against humans, and if they are also toxic against humans, that means they are basically useless compounds. So, Pasteur's theory remained as a theory, it was never practically implemented. Now, in the year about say 1900, what happened? Another a scientist whose name was Paul Ehrlich. Paul Ehrlich's picture is here. He is working in a lab, and by the side of him is a Japanese scientist. His name was S. Hata. Okay. So, Ehrlich was the first one to believe that it is possible to develop chemotherapeutic agents. In fact, the term chemotherapy was coined by Ehrlich, Paul Ehrlich. So, Paul Ehrlich thought that it is possible to develop chemicals which will, if we take that inside the body, then it will target the causative agent for the infection. That means, if I am suffering from any bacterial infection, then Ehrlich believed that there is a possibility that you can design chemicals which, if it is ingested, then that will not touch the host machinery, but it will go and attack the foreign organism, and then it will kill that, and that he termed as magic, bullets. So, magic bullets are nothing but chemicals, but it is why it is called magic? Because it has got a, it does not affect the host, only thing that it affects only the microorganism from which is the causative agent for the infection. So, these are called like bullets, you target to a particular person and then you kill that person. Now, in the body you are not seeing what is where are these bacteria, but it is possible to create magic bullets which will find the bacteria and kill it. So, that was his theory, and these magic bullets are nothing but he called the chemotherapeutic agents. So, he prepared different compounds, several compounds, thousands of compounds he made on those days, and he found, both of them found that this compound number 606. So, every compound has a number, compound number 606 is a very good antibacterial agent. Okay. And this he commercially, it was named as Salvarsan. So, Salvarsan, I think A last. Salvarsan is the compound, the first known antibacterial agent. It is based on, it is an arsenic based compound, Okay. And in very soon Ehrlich was awarded the Nobel Prize in Physiology and Medicine because of his discovery of Salvarsan, that is the compound number 606. And then subsequently in our country, Sir UN Brahmachari in Calcutta, he discovered a molecule which was called urea stibamine, which is now an antimony based stibamine. That is an antimony based compound, okay, SB, antimony based compound. It was well known those days that arsenic, antimony and bismuth, they are having antimicrobial activity. So, people were starting to make these arsenic based compounds, antimony based compounds and bismuth based compounds. In fact, all the three are utilized to develop molecules which are drugs. So, Salvarsan is an arsenic based compound and UN Brahmachari's compound was antimony based compound. This was an antibiotic, antibacterial agents, because there is a difference in antibiotic and antibacterial, I will tell you later. Arsenic based compounds, and it treated a deadly disease called syphilis, which was untreatable in those days. And antimony compound, this urea stibamine, was utilized or was used for treatment of black fever, which in Bengali it is called kala jar, okay, black fever. So, these are the initial developments in the history of drug discovery, the antimicrobial drug discovery we are talking about, okay. Because before that, this your this aspirin was discovered, that was at the late 19th century. So, this is early part of 19th century. And however, because these are arsenic, antimony and these are these metal based compounds, anyway these are to be very toxic. These were later on found to be very toxic. So, apart from their beneficial action, they also have extreme toxicity. So, these especially this compound, this arsenic salvarsan, is now no longer used because of its toxicity. So, people started to look for different compounds. So, people although were killed of syphilis, but they might be killed by the toxicity of the arsenic based compound. So, which is not desirable. Then came in 1928, when Fleming discovered the penicillin, and exactly what happened, that Fleming had all these are all his petri dishes, because antibacterial activity are determined by taking a covered plate like what is called a petri dish, and you grow bacteria inside the dish, and you see the effect of different chemicals on the growth of the bacteria. So, what Fleming observed? Fleming observed that one of his plate, which was lying on his bench for quite some time, because Fleming went for a holiday, and 2 weeks later he came back, and without cleaning his petri dishes. So, he found one of his petri dish, he looked at it, and he found that if you look at this, this is there is a growth of a fungus, and around the fungus there is no growth of any bacteria. This is a clear zone. So, the bacteria was growing like this, because wherever there is a haziness, that indicates that bacteria is growing, but around this mold, this fungal mold, nothing is, no bacteria can grow. Okay. So, Fleming immediately realized, because he had a brilliant mind. So, he immediately realized the potential of this mold. And interestingly, what happened, that Fleming was working on the first floor of what is the Queen's Mary Hospital, and in the ground floor there was another colleague of him was working with different fungal strains, and one of them was Penicillium notatum, that is the fungus, and these Penicillium notatum fungus, they flew from the lab at the downstairs at the ground floor, went out of the window, and finally came through the window of Fleming's lab, and it is just extremely sheer luck that that fungus went inside the petri dish. Okay. Because petri dish, you have to open it and then you have to close it. Na? When you put the bacteria, you have to open it, and then you have to, after spreading the bacteria, you close the lid. Okay. So, that time that fungus went in and went inside the petri dish. Okay. So, there are many stories, everything. This is a serendipitous observation, that is true, but other people might have just washed this plate, but Fleming, while checking all the plates, he found that the plate which he was going to wash, clean and autoclave, but he found this miraculous observation, and that gave the birth, that is the birth of penicillin. Now, what happened then penicillin, then Fleming tried to grow this microorganism, this fungus Penicillium notatum fungus. He grew in flask and tried to isolate the compound, that means the compound which is secreted outside this fungus. He tried to isolate. He named the active compound without isolating it as penicillin, because it is being produced by Penicillium fungus, but he could not isolate penicillin. But he found that the crude extract of the fermentation of this Penicillium fungus, if you grow it and take the filtrate, that filtrate had lot of antibacterial activity. Okay. But he could not isolate it. So, it had, along with the active compound, it had other compounds in it. He tried to work on mice, that what is the toxicity level. You know, all drugs need to be shown that they are not toxic. So, toxicological effect was fine, but because the active principle was present in tiny amount and he wanted to isolate it, then he found that it is very unstable. He could never isolate the penicillin. So, he gave up the research, just publishing 2 results, one in British Journal of Pathology and another publication later on in 1932. The first one is 1929 and then the 32. So, these 2 publications were forgotten by the scientific world, but later on it was 2 chemists at Oxford University, Howard Florey and Chen. They again went back in 1939 and took the fungus out from Fleming's laboratory, made Fleming, got the fungus and then grew it, and then they isolated the penicillin and tried to determine its structure. Some proposal was there that this is the structure, but finally, what happened, the structure was confirmed by Dorothy Hodgkins, the famous crystallographer, women crystallographer in Oxford. So, he finally proved the structure of penicillin. So, that is the story and that is the beginning of the antibiotic era. Now, let me say what is the difference between antibacterial agent and antibiotic. Antibiotics are compounds which are produced by microorganisms and it is for their own benefit, for their own survival to kill other surrounding microorganisms. So, antibiotics are produced from microorganisms, whereas antibacterial agents are a broad class of compounds, any compound or any chemical which is having a power of destroying any microorganism, that is called antibacterial agent or if it is bacteria, say antibacterial agent. However, the general term should be antimicrobial agents, because antimicrobial will include everything: antiviral, antibacterial, antifungal. Okay. So, this is the difference. So, basically antibiotic are basically they are isolated, they are produced by microorganisms. Okay. So, we do not have to do much, we just isolate, grow the microorganism and start isolating the active principle, and there are many compounds which are in the market, they are all antibiotics. Okay. Now, let us again just quick revisit. Chemotherapy is basically the use of drugs that treat a disease and a drug that kills harmful microbes without damaging the host. See, chemotherapy means that means it has got a selectivity, selective toxicity. All compounds are not chemotherapy, are not branded as chemotherapeutic agents if they have high selectivity, high toxicity. It is only that chemotherapeutic agents not only treat the disease, but at the same time it should have less toxicity in the animal or human where it is being prescribed. Okay. It is again the same thing I just already told. What is an antibiotic? Chemicals produced by a microorganism that kills or inhibits the growth of another microorganism. An antimicrobial agent is basically a chemical that kills or inhibits the growth of microorganisms. So, I can say that all antibiotics belong to the class of antimicrobial agent, but all antimicrobial agents are not antibiotics, because some could be synthetic compounds. Okay. Now, these are some of the different antibiotics that have been isolated from different sources, different microorganism sources. I will say this penicillin I have already told you, that has been isolated from Penicillium notatum. Grisofulvin is an antifungal compound which is isolated from another Penicillium sources. Cephalothin is a compound which is isolated from Cephalosporium species. Gentamicin, these are very well known: streptomycin, erythromycin, chloramphenicol means chloromycetin, and then amphotericin B, and interestingly many of these are associated with Nobel Prize winning work. Okay. So, this is a whole lot of compounds that have been isolated from microorganisms, and there is no end to it, because if you today you want to isolate, if you want to enter into this field antibiotic, what you have to do, you have to see the different new microorganisms you have to isolate, and then see whether what type of chemicals it is generating. However, the multinational companies, the pharmaceutical companies, it is hard to beat them, because they are having a collection, a huge library of microorganisms, and then try to see what type of compounds they produce. So, usually this is in the domain of pharmaceutical industry and not academic. Academically it is very difficult, because you can only touch few of the microorganisms, but this is not the way. This is the same thing, like combinatorial library of different compounds gives you a possibility of identifying the heat quickly. Similarly, a library of library of different strains will be much better, has a better chance of success of getting a new antibiotic compound. Okay. These are some of the spectrum of activity of the antibiotics and other antimicrobial drugs. Like, see, we suffer, there are different agents that cause diseases, like mycobacteria, which is the causative agent of tuberculosis and leprosy. Okay. Then you have this prokaryotic bacteria, prokaryotes, and between the prokaryotes you have gram negative and gram positive. Okay. That is the gram sensitivity to gram reaction. Then you have there are other types of chlamydias, that could be that fungi, means fungal infections, which are actually eukaryotes, that is the higher organisms. Protozoa is the malaria one. Then helminths mean, actually this is from the tapeworms, that people suffering from this worm infection. So, these are little bit longer in one direction, and then you have viruses. So, this is the spectrum of different different organisms that we suffer from, and there are different kinds of, remember, no antibiotic is effective against all the microbes. It is not that you have one microbe that is going to kill all these, from fungi to virus to mycobacteria, that is not possible. Mycobacteria, that means tuberculosis drug, one of the earliest drug is streptomycin. For gram positive bacteria infection, penicillin was the drug of choice. Okay. Then you have mefloquine, that is for malaria. You have tetracycline, which has got a broad spectrum. See, you know, narrow spectrum antibiotic, broad spectrum antibiotic. Broad spectrum means which actually covers many of these microbes. Like tetracycline is a broad spectrum antibiotic, which is broad spectrum antibiotic, and the penicillin, some of the penicillin are quite narrow, they just kill gram positive bacteria, but we will cover that how the penicillin, there are now different types of penicillins which can also kill gram negative bacteria, but gram positives are the real targets of penicillins. Then isoniazide is a synthetic compound that works against tuberculosis. So, that is the spectrum. So, when we talk about antimicrobial chemistry, so we have to touch this prokaryotes infections and the eukaryotes infection and the viruses. Okay. But we will take we cannot actually cover all these in this course. We will just take some examples which have become the landmark of discovery of antibiotics. Okay. Now, mechanism of antimicrobial action. Remember, microbes have a life. So, they have a life cycle, and they also utilize some of the processes that are also inbuilt in our that are also going on in our body. However, there is a difference, there are some differences between the enzyme systems in the microbes vis-a-vis the humans. Okay. Like bacteria have their own enzymes which is not present in the host. Host means I mean the animal, the human, etc. Like cell wall formation, we will talk about that. What is this cell wall? That means, in bacterial cell there is a wall surrounding the cytosol, that means, whatever the components inside, that is protected by the wall. Then protein synthesis, we also have protein synthesis, we have already read this replication, transcription and translation. However, their enzymes might be slightly different from the enzymes that we have, okay. And if that be the case, then you can exploit that difference. Remember, when we talked about the folic acid chemistry, we encounter an enzyme which is called dihydrofolate reductase. This is present in bacteria, this is present in human, but the dihydrofolate reductase is different from in a microorganism versus the human. So, you can selectively target the DHFR of a bacteria. DNA replication, see, these are all processes which are common to the host, RNA synthesis, transcription and DNA replication, this is translation and synthesis of essential metabolites. However, these can be the targets for devising an antimicrobial agent. Okay. And the reason is that I said that although some of the enzyme systems are common or processes are common, but there is a difference in the enzyme structure, primary structure, that means the amino acid, and finally, the tertiary structure is also different. So, you can exploit that difference. Okay. I think we will talk about this, that how you can develop antiviral compounds, antifungal, and but this is very important. Whatever may be the target, the more similar the pathogen, pathogen means which is causing the infection, the organism and the host enzymes, if the similarity is more, you are going to have more side effects, and if the similarity is less, then you will have less side effects, and if there is no similarity, then you will not have you can expect that there is very few side effects. Okay. In fact, penicillin is one of them, that it has got extremely less side effect, okay, because it targets an enzyme which is not present in the human system, which is not required in the human system. Now, this is a schematic diagram. I told you what are the targets earlier, but this diagram makes it a little bit easier to understand, that this is the bacterial cell, and in the bacterial cell there is this DNA, and you have this transcription process, mRNA goes to protein, and what you also have, and the protein makes different small molecules which are essential for the survival. Now, where you can target, which can be the target for your drug discovery process, which are antimicrobial agents. One is cell wall, because the cell wall is not present in the human cell. The human cell is protected by what is called a membrane, a lipid membrane, that is human cell. Okay. In the bacterial cell, there is, apart from the lipid membrane, outside there is a coat, which is a very rigid type of system, and that coat is extremely important, because inside the bacterial cell the osmotic pressure is very high. Osmotic pressure is very high, Okay. If the osmotic pressure is very high, so that means they are pushing they are trying to go out. Okay. And so, outside is osmotic pressure low, that means, when I put the cell in a biological medium, outside the osmotic pressure is low, inside is high. So, what happens? The water from outside wants to enter into this cell, but in order to prevent that, because if water goes here, then that has to swell, because it has to accommodate the water, and there is basically no space. Already it has got a lot of cytosolic contents inside, which creates this high pressure. So, as water goes inside, it swells and then it bursts. Okay. But the question is, how water will go inside? So, what you do, you create cavities, that means, you make molecules which will not allow the bacteria to form this cell wall. So, one good target is cell wall, and this cell wall we do not have. So, cell wall synthesizing enzymes can be very good targets for antibacterial agents. Okay. Then you have inhibition of protein synthesis. So, there are molecules like chloramphenicol, erythromycin, tetracycline, streptomycin. What they do? They stops the translation process. Okay. This cell wall is inhibited by penicillin, cephalosporins, bacitracin and vancomycin, a very important antibacterial agents, life saving antibacterial agents. Then you can stop the transcription process also. Transcription process, you have quinolones, like you must have heard of ciprofloxacin, norfloxacin or rifampicin, that is that is again a tuberculosis drug. They stop the transcription process. So, you can stop the production of mRNA by these quinolones. You can stop the translation process by chloramphenicol, erythromycin, etc. You can stop these enzyme activities making of important small molecules like your folic acid, where this you can take sulfonamide. Remember, sulfonamide is a PABA analog, and then trimethoprim is a dihydrofolate reductase inhibitor in bacteria. So, all these are the targets: bacterial cell wall, translation machinery, transcription, and then you can have functional aspect where the protein which synthesizes different small molecules, and you can stop somehow the small molecules, like dihydrofolate reductase was one example. These are some of the structures which are isolated from the microorganisms. Okay. This is the structure, general structure of penicillin. See, these structures, they have only similarities between this and this. This is the cephalosporins and that is the penicillin, but other structures are entirely different. This is called rifampicin. And this is what is called tetracycline. You do not have to remember the structures. My own only intention is to show the variety of structures that these microorganisms make, including stereochemistry. Everything is very stereospecific. This is your ciprofloxacin. And this is the chloramphenicol. Okay, so these are the, but apart from that there are other molecules which have again another different types of structure. We are going to cover penicillin, we are going to cover cephalosporin, they come together, and we will also discuss some of the mechanisms of possibly chloramphenicol if time permits, ciprofloxacin, those we will talk about that what basically they do. So, now another important, I have already told you what is the difference between antimicrobial agents and antibiotics. Okay, that is very clear. Another term which has to be made clear is bacteriostatic and bactericidal. Bacteriostatic and bactericidal. Okay. Actually, antibacterial agents, bacteriostatic and bactericidal. Okay. What is bacteriostatic? Bacteriostatic is basically that if somebody is suffering from some bacterial infection and you add some chemicals, some drugs which stops the further growth of the bacteria. Like if a bacteria is already there, say 100 bacterial cells are inside my body. Okay. Now, what they want to do? They want to divide, 100 will be 200, then 200 will be 400, but for the division to occur you have to utilize the machinery, the replication, transcription and translation. Now, there are molecules which act, I already told you, chloramphenicol, which actually stops the protein synthesis by inhibiting the translation machinery. So, what will happen? The existing cells will remain because they are already matured, but when they try to divide, that is not possible. That means, I will have these 100 bacterial cells, I will not kill them, Only thing what I will do, I will not allow to grow them. So, that is what is called bacteriostatic. That means, the bacterial population remains the same, but then the question is, how do I cure myself? Because if the bacteria does not grow, that gives enough time for the body to make antibodies, and then antibodies ultimately take care of this whatever microorganisms are there. Okay. So, that is bacteriostatic. On the other hand, compounds which are called bactericidal. Bactericidal means they go and kill the existing bacteria. That means, the bacteria 100 bacteria will go to nearly 0, almost 0. So, you will see the difference. In one case, you are killing the bacteria, bactericidal, and penicillin one example, which kills the bacteria, and bacteriostatic is which does not kill the bacteria, stops the growth. Both are antibacterial agents, but definitely if somebody has very weak immunity, like somebody who is suffering from HIV infection. HIV is a viral infection which lowers, which ultimately attacks our immune cells and breaks down the immunity. So, if he is suffering from an HIV patient suffering from infection, bacteriostatic will never work, because he does not have the immunity. So, how can he take care of the remaining whatever remaining population of bacteria there? Or even in transplantation, say kidney transplantation, what happens? Your external that foreign kidney is given from the donor to the acceptor. However, because it is a foreign, it is not it is not a part of the body system from the very beginning. So, the body thinks that it is a foreign agent, and whenever body thinks something is foreign, it generates antibody, and then that means there is an immune response, and that rejects the. So, there are many cases where the kidney transplantation is not successful, because there is this body does not accept the foreign kidney as its own. So, what is usually practiced, that at that point kidney donation, you lower the immunity of the body practically to very low level, and then you do the transplantation. So, at that point you slowly increase the immunity. So, immunity was very low, slowly you increase the immunity, and ultimately say after 3 months, 4 months, the body thinks that it is my own material. Okay. So, at that time if there is an infection, you cannot treat that person with bacteriostatic drug, you have to give bactericidal. Okay. So, that is an important difference I told you. I think in the next session we are going to start with the chemistry of penicillin. So, it is a very background introductory remarks of antimicrobial compounds, and then we will slowly go to the actual drugs, their mechanism, their discovery, and all these things. Thank you.